Stenotrophomonas, multophilia. And you can find this in both the environmental and clinical settings. So it follows in line with the topic that we're going to be talking about in One Health today. Okay, so in terms of clinical significance of S. multophilia, currently the WHO lists this as one of the leading drug-resistant organisms worldwide. Two antimicrobial surveillance programs have listed the S. multophilia as one of the top 4 pathogens. How's that? Oh, that's much better. That's much better. Now I can back off from it. Okay, all right. So as I was saying, the 2 antimicrobial surveillance programs, one in 2012, the other one in 2012, list S. multophilia among the top 10 pathogens in both of these studies. Big note about S. multophilia, it is an opportunistic pathogen. So it's a significant concern in those patients that are immunocompromised. So patients that are, say, taking cancer chemotherapy drugs, or patients that have long-term hospital stays and associated with indwelling medical devices. So this is an organism that has a high mortality rate associated with it, as high as 70% in those that are untreated. Okay, this is a TM micrograph showing S. multophilia. It is a gram-negative rod. As you can see, it's got several polar flagella sticking out of it that help it in terms of motility. A closer look at the surface on some strains will reveal the presence of fimbriae and pili. So where you can find S. multophilia, again, I said you can find it in both the environmental and the clinical setting. In the environment, you can find it pretty much anywhere there's water associated with the surface. So in freshwater settings, animals, for the vets in the audience, this is becoming an organism that is becoming more and more significant for veterinarians treating infected animals with this pathogen. You can also find this in the soils, plant rhizosphere, washed foods, and then in the clinical setting, which is where I'm primarily going to talk about today. You can find this associated on medical devices, implants, particularly catheters, indwelling catheters, nebulizers, faucets, moist surfaces such as shower heads, sink drains. You can also find steno. Interestingly, for the dental conference that's going on next door, you can also find S. multophilia forming biofilms in the dental suction line. So this is actually the tubing that goes into your mouth when you go visit the dentist every 6 months. So I'm talking about that particular tubing. Interestingly, you can also find S. multophilia in soda machines and in the sink drains in both the clinical and environmental setting. Again, this is an organism that is found anywhere there's really a moist, supportive environment for it. If you want more information about S. multophilia, where it's found, I published in 2012 a review in the Clinical Microbiology Reviews Journal. And to give you an idea of this pathogen, when I first started in 2001, you can pretty much count on 10 fingers how many papers are actually coming out about this organism. Since then, it's just skyrocketed in the actual literature. And this review of mine has actually been cited over 200 times already. Okay, so this is some work that was done in my lab. It's published in the Journal of Environmental Health. What we did in this article, we were looking at different surfaces in a non-clinical setting and asking the question whether Stenotrophomonas could be found. And it turns out that yes, you can find this in sink drains on urban college campuses. So it turned out in one particular study that we did, over 60% of the sink drains actually contained S. multophilia. So you can find it in the biofilms present. In terms of the microbiology of this organism, I've said it's a gram-negative bacillus. It is an opportunistic pathogen. If you look at it growing on blood agar, you can see the absence of hemolysis, so we say this is gamma hemolytic. It was formerly known as Pseudomonas multophilia. It was Anthomonas multophilia. So 16S ribosomal RNA sequencing made it be reclassified into its own genus Stenotrophomonas. A few other characteristics: biochemistry, it's oxidase positive, catalase positive. It has an unusual feature of being able to hydrolyze esculin. Many strains produce DNAs, and as I mentioned, it's motile. One of the nice things about looking for Stenotrophomonas in the environment, you can grow this organism pretty easily. And on McConkey agar, it's lactose fermentation negative, so it's pretty easy to pull this one out from, say, Pseudomonas strains. Okay, so one of the criticisms about S. maltophilia, one of the big discussions that's still ongoing to some extent, is whether or not S. maltophilia is simply a colonizer in terms of infection, or whether it's a standalone pathogen. And the reason is because this is a common drinking water inhabitant. So is it really causing disease, or is it just a colonizer that happens to be present in infection? It's associated with various human infections, most commonly of the respiratory tract and then of the bloodstream. You can also find this in the brain, heart, bones, soft tissue. The risk factors for the infection are the presence of indwelling devices, long-term use of antibiotics, long-term hospitalization, stay, and chronic respiratory disease, and of course being an immunocompromised host. There have been reports of both nosocomial and community-acquired infections with this organism. It is a co-colonizer. This means you can identify it in infections involving both gram-positive pathogens, such as Staph aureus, and gram-negative pathogens, such as Pseudomonas aeruginosa. One of the most common incidences is to look for Stenotrophomonas in the cystic fibrosis lung. It's part of the cystic fibrosis flora. We do say that this organism is of low virulence. It has a low ability to invade cells as well. In terms of the studies to dissect out whether it's a colonizer or a standalone pathogen, the earlier studies using mouse infection pneumonia models saw that there was a high inflammatory response. This was done by two groups, Valerie Waters, Giovanni Di Bonaventura. Later in 2011, Valerie Waters came out with a really nice study where she looked at cystic fibrosis patients and asked, was there a specific immune response to Stenotrophomonas? And yes, there is. Turns out that patients are mounting a definite antibody response to these flagellum that's found on the surface of the cells. So they're saying that in the infection, this organism is causing direct infection in these cystic fibrosis patients. There's a direct immune response to Stenotrophomonas. Okay, one of the problems with working with Stenotrophomonas, it's multidrug-resistant. You can see there's many different types of drugs that it's resistant to intrinsically. This means there are many genes found on the chromosomes that provide resistance to these different drugs. So there's a definite need for new drugs. What else is new about these pathogens? Or alternatives, and I write about this in an editorial here, if you're interested to go and read that. Okay, so in terms of in vitro antimicrobial susceptibility testing, this relates to one of the comments you made earlier. We have to get away from thinking about planktonic cells and start thinking about biofilms. This is where steno is found mostly. So one of the problems with the antimicrobial susceptibility testing is that if you look around the world, there's no standardized procedure that's being used. So we've got different tests being done, e-tests, distillation tests, different standards being used depending on what country you look at. So there's a need to get standardization of testing being done. That's one. The other thing is, right now, we just have MIC breakpoints available for some antimicrobial agents, not all of them. So again, it makes it difficult assessing where's the acceptable level to use in terms of these drugs. This is the main part I want to get to here. We need to start thinking about biofilms. This is a respiratory tract infection organism. So a lot of our antimicrobial susceptibility testing relies on the drug concentration we can achieve in the bloodstream. That's fine. But we're talking about an organism that is part of a biofilm in cystic fibrosis patients. So we also need to think about testing in terms of what kinds of levels of antibiotics can we get into those patients if it's delivered in an aerosol form. Okay, so we need to establish guidelines for CF patients with respiratory secretions. Guidelines need to be established for isolates from patients with chronic S. maltophilia infections. In terms of genome sequencing, what I did with this slide is I focused really on the clinical microbiology side of Stenotrophomonas. I mentioned you can find strains in the environment. One of the earliest studies to look at the genome sequence looks at this strain. This is a reference strain, K279A. This has come from a bloodstream infection of a CF chemotherapy patient. The sequencing of this revealed many multidrug efflux pumps. The R&D type is the most common type studied. It turns out in Stenotrophomonas, there's about 6 different families of these efflux pumps. And they're responsible for resistance to things like quinolones, chloramphenicol, tetracycline, a multitude of drugs, and also toxic compounds. Also in the genome sequence, we found the presence of transposons, insertion sequence elements. This suggests there is possibility of horizontal gene transfer going on between these different isolates. I put these 3 up just so you can see the sequencing is ongoing. More and more clinical isolates are being sequenced. What is interesting is the common theme running through these sequences when you look at them is the presence of these multidrug efflux pumps. So this is providing most of the intrinsic resistance to drugs in this organism. So genetic diversity among the strains, yes, it exists. We use PCR-based fingerprinting, PFGE, analyses to try to look at similar grouping or similar clonality among different isolates. There are new sequencing methods that are becoming available to increase the rate at which we can sequence these genomes. I mentioned the intrinsic drug resistance, mostly due to presence of chromosomal genes. The MDR phenotypes, we're seeing a lot of variation in these. And some of this is due to normal evolution, accumulation of mutations. And some of this is being shown to be due to the presence of antibiotics. So there's a selection pressure there that's causing a change in the MDR phenotype. Yes, there's a possibility of horizontal gene transfer in S. maltophilia. Keep in mind this is an environmental pathogen. So I'm talking about gene transfer in the natural habitat, in the soils. It's been shown by conjugation, for example. You can have transfer of plasmids between S. maltophilia and even other genera of organisms. In CF patients, there's a really interesting study. It's come out fairly recently, in 2014. What this study looks at is it looks at CF patients, and what they're doing is they're looking at how many genotypes are present of S. maltophilia. And they also looked at specific genes involved in mismatch repair. And they asked the question, what's the mutation rate like over time? And it turns out that the mutation actually changes from being highly mutable rates to low mutable rates. And what it's suggesting is that the strains themselves are adapting themselves to the environment in which they're found. So it's a way of kind of preserving their genome, not accumulating too many deleterious mutations. Okay, so in terms of mechanisms of drug resistance, this is a busy slide. Essentially, you can find this in the review that I wrote in Clinical Micro-Reviews. What I did here, to be fair, because there are a ton of references, not just these, I tried to acknowledge the people that did the early work, and I tried to look forward and see who is publishing the most recent work. Just by looking at the slide, you can see there's a lot of work being done in this area. This is a hot topic in terms of steno research. So, in terms of drug resistance, the top 2, beta-lactamases, reduction in outer membrane permeability, provide resistance to the beta-lactam drugs. Class 1 integrons, insertion sequence, common region elements, provide resistance to things like the sulfa drugs. Multi-drug efflux pumps I've already mentioned. What I will say is this. There's some nice work done by Gabriella Berg's group where they've looked at Stenotrophomonas strains in the natural environment, in the soils, for example, and they see the presence of these multi-drug resistance pumps. And the idea is to thinking that it's not the acquisition of these strains into the hospital environment and then somehow magically they picked up these efflux pumps. These efflux pumps naturally exist in those strains found in the environment, so they must have some other function other than just extrusion of drugs. So it's been shown that one of the pumps in particular is responsible for the adhesion of the bacterium onto a plant surface. And there are other types of functions that can be looked at in terms of these drug pumps. In terms of LPS in Stenotrophomonas provides resistance. There's one particular gene, SPGM, that encodes phosphoglucomutase. Turns out if you make a deletion in this, you actually increase the sensitivity of this organism to a particular drug. There are also SMQNR determinants and mutations of bacterial topoisomerase and gyrase that are responsible for resistance to quinolones. And finally, there are some enzymes found in S. maltophilia that modify drugs such as the aminoglycosides. So you can see there's a whole plethora of molecular mechanisms that work here in Stenotrophomonas. So one of the other mechanisms of resistance to drugs has been the ability of this organism to make biofilms. So it makes biofilms on medical devices, implants, dental tubing lines, various abiotic surfaces. I'm told by one clinician that I work with that you can have the situation where the biofilm is so extensive on the catheter inside the patient that the only thing that's left, because it is so multi-drug resistant, is to simply remove the catheter and hope that the patient's own immune system can fight off this organism. It can also form biofilms on living tissues. This is just an example showing you what the biofilm of Stenotrophomonas looks like. This is on a polyvinyl chloride microtiter well using the standard crystal violet assay. Okay, so some of the early work done with the biofilms is looking at the cell ultrastructure, trying to figure out which components of the Stenotrophomonas are important for biofilm formation. Some nice, elegant ultrastructure work was done by Oliveira Garcia et al., 2002-2003. Essentially what you're looking at is a clinical isolate of S. maltophilia sticking to HEP2 cells, living cells, and also to the plastic surface. They made the point in these two papers that these Stenotrophomonas rod-shaped cells have flagella that help stick and have fimbriae, and they proposed that the fimbriae would be important in biofilm formation. Later on, it looks like this is of less significance, but I wanted to point it out that it had actually been looked at. In my own lab, with my students, we used transposon mutagenesis on Stenotrophomonas. And we made what's called a FlyF mutant. Essentially what you're doing is you're knocking out the flagellar expression. So these cells lack flagella. This wild type has the polar flagellum attached. You do the crystal violet assay, you look at biofilm formation, you see there's no real significant difference between having flagella and not. So it looks like the biofilm is independent of the flagella. And this is also confirmed by work in Giovanni di Bonaventura's group, where they looked at flagella, motility, adherence, and invasion of human cells with CF isolates of steno. Again, couldn't see any real correlation. Okay, so moving on to LPS. This is also work from my lab, where again we're using transposon mutagenesis. This time we're making an SPGM mutant. Essentially what this does is it doesn't change the chemical composition of LPS, but what it does do is it lowers the expression of the high molecular weight LPS. So this is actually an SDS page gel of the lipopolysaccharide. As you can see over here in this other diagram, the biofilm formation of the SPGM mutant is quite rapid, within 4 hours, and there's a lot of it. So it suggests that Decreasing the length of LPS actually lets the cells come into closer contact with the surface very quickly, and so you get a lot more biofilm as a result. Okay, S. maltophilia is biofilm. Yes, it's influenced by culture conditions. Again, in my lab, we looked at phosphate levels. If you increase or decrease the phosphate, you can increase or decrease the biofilm. pH and temperature was looked at by the other group, Di Bonaventura's group. There's optimum temperatures for biofilm formation. Turns out biofilms are more established under aerobic conditions than anaerobic. And it turns out there's no difference between static and agitation in making these biofilms. What is very interesting to me is this. And this relates to, I guess, what we were talking about earlier, about thinking big picture here, how these organisms are interacting. So it turns out in S. maltophilia, there's something called a diffusible signal factor quorum sensing mechanism. The signal factor itself is a fatty acid. This has been well established for quite a while now. What is interesting is to look at the recent work that's just come out. This is a very, very interesting study. To get the chance, pick up this paper and have a look at it. What they did was they teased apart two populations, two variant strains of Stenotrophomonas, and they show that one strain is lacking the ability to make the DSF. And that DSF mutant, this RFP2 variant strain, actually does something called social cheating, which means it comes in contact with the RFP1 variant strains and starts making DSF. It's a great way to conserve its energy without having to make DSF until it runs into the RFP1 variant strain and starts making it. And that's interesting in itself. What is even more interesting is they combine these two populations and they show that they can work synergistically. And they can increase the mortality rate almost 100% in the adult zebrafish infection model. So this, I think, is very interesting. It's telling me that the cells are able to sense each other. They're able to talk to each other. Okay, that's Stenotrophomonas alone. Now taking one step further, and look at what's happening in the CF patient. Again, you've got a multitude of organisms present. One example is Pseudomonas aeruginosa. So it turns out that DSF is actually present in the sputa of CF patients. Okay, so it's present. Turns out that DSF also allows the persistence of Pseudomonas aeruginosa in these patients. So it's telling me there's some sort of interspecies interplay between Stenotrophomonas and Pseudomonas in the cystic fibrosis lung. They're able to talk to each other. Very, very recently, 2015, not up on this slide, run across a paper that says Stenotrophomonas can receive signals, acyl homoserine lactone signals from Pseudomonas. So there you have it. We do have interspecies signaling. It's a biofilm. So this relates to how I'm going to treat this. Because the presence of DSF for Pseudomonas aeruginosa is very helpful. It changes the Pseudomonas biofilm, changes its architecture, and it makes it more resistant to antimicrobial peptides. So it's making the biofilm more resistant to polymyxins. So again, it's Stenotrophomonas helping out Pseudomonas, those two working together. So I'm going to show you just real briefly this alteration I'm talking about with the Pseudomonas aeruginosa biofilm. So this is elegant confocal microscopy done by Robert Ryan in 2008. I'm just going to orient you to two panels. So they are using Pseudomonas aeruginosa PA01 standard strain to look at the biofilm. This is it. It's fairly flat monolayer. It turns out when you give this Pseudomonas exogenous DSF produced by the Stenotrophomonas, it changes that biofilm quite significantly. It goes from that flat monolayer to this filamentous architecture. Keep in mind, this is less resistant to polymyxins than this is. So again, these two organisms are talking to each other. In terms of strategies for treatment, currently the trimethyltrim sulfamethoxazole is the #1 line of treatment. But as with all the other drugs we've been talking about, there's emergence of resistance. So we have to start looking for alternatives. Alternatives may be in the form of fluoroquinolones. One example is levofloxacin. There are others. But again, resistance is rising. Tigecycline, a member of the tetracyclines, may be an alternative, but there are studies out there that look at the dosage level given to human volunteers, and there are some adverse effects we have to pay attention to when the concentration levels get so high. So again, that might be of concern there, delivering this drug. Early work by John Lapuma's group, 2009, suggested we could use nanoemulsions. These would cause the outer membrane of Stenotrophomonas to fuse, therefore killing off the cell. So I can talk to you a little bit more about these. There's not a lot of work coming out about the nanoemulsions now, and I think partly it's due to patents that have taken place. There's a lot of interest in antimicrobial peptides. Okay, terrific. Antimicrobial peptides are of interest, so a lot of these are coming from amphibians. And we're looking at these because the most attractive feature is their ability to to penetrate the outer membrane quickly, kill off the organism. And the big advantage is they have lower rates of resistance than antibiotics. So this is an area that's pretty hot. Finally, we'll talk about the phage therapy. This is extremely exciting. Go check this paper out. It's by Peters et al., 2015, just come out. This paper is the first paper I'm aware of where they're showing 2 different phages against Stenotrophomonas. Okay, 2 different phages that can infect some strains of Stenotrophomonas and infect some strains of Pseudomonas. It's unusual to have phages cross genera boundaries like that. This is very exciting. This could be one way of getting around the antibiotic resistance problem. In my next talk, which is in a little while, I'll talk to you about a combination therapy that I'm trying to look at, see if it's got some use. Okay, I am promising you I'm concluding really quickly. Just a plug for those of you that might be interested in Stenotrophomonas now that you've heard my talk. I'm actually one of the editors. I'm the lead guest associate editor with Frontiers in Microbiology. We've got online right now a research topic on steno. What's nice about this is you can also find out information about the environmental side of steno. I chose to focus on the clinical side today. Finally, I'd like to acknowledge funding from my lab, which has come from NIH, internal funding from DePaul University, and most importantly, my collaborators and my undergraduate students. You notice it's a pretty short list. These are the ones I've had over the past year and a half. I've had many, many more undergraduates. I just simply can't fit them on the screen. These are my MS graduate students who have all had a hand in working with Stenotrophomonas. Thank you very much for your attention.